So before returning to heat treatment, I realised over lunch that I skipped over inadvertently a piece of information that I should have given you earlier. It is about Qatar and its anti-hepatitis B testing. That testing continued until the end of October 1984. It was discontinued at that time, mainly because it was stated that there was sufficient confidence in the heat treatment process at that time to allow for the discontinuation of the anti-hepatitis testing. It was also noted in the internal company documents that a specific test for HTLV3 was expected shortly. That's at paragraph 203 of the written presentation. Moving back to heat treatment, on the 30th of May 1985, a meeting took place between the FDA and fractionators about the question of what, if any, regulatory action should be taken over non-heat treated products. We can have on screen, please, BAUM 5025. Please ignore the highlighting on this document. That's not ours. The date of the document is the 30th of May 1985. It's from Dr. Ujala. As we know, Dr. Ujala is a regular source of our information for such meetings, and it's an internal Qatar memorandum. The memorandum says, Dr. Harry Mayer, who's a senior figure within the FDA, called this special meeting of all producers of coagulation products to discuss the use, production, license of non-viral inactivated products. And non-viral inactivated products we take to mean heat-treated products. The attendees included...
No, not heat-treated products.
Sorry, yes, not non-heat-treated products, yes. Thank you. The attendees included Dr. Pistriciani and Dr. Donahue from the FDA, as well as a number of other members of the FDA, and the figures from the industry that we've become accustomed to seeing at these meetings, Dr. Ujala, Dr. Holst of Highland, Dr. Carr of Alpha. From ARMA, there's a D. Marcus, rather than Dr. Riddell, who is usually present, and also representatives of Host Immuno, the American Red Cross, and Bob Riley of the American Blood Resources Association, a trade body. If we go back to the text, Dr. Mayer explained that the major manufacturers of coagulation products, AFA, AHF, and PTC, have been approved for a viral inactivation process for some time, and the data demonstrated reasonable performance for eliminating HTLV3 virus from the final product. He questioned the utility for a non-treated process given the current situation and requested that we uniformly send letters to the FDA stating we would no longer produce or distribute non-heated product to preclude negative reaction from the medical community and the general public. He indicated that everyone spoke as though heat-treating processes had eliminated the potential for HTLV3 viral exposure from these products, and no one had really focused on the fact that the ability to produce non-treated products still remained possible with the current licenses. He explained that although the FDA could revoke these through the regulatory process, he did not want any attention paid to the fact that the FDA had allowed this situation to continue for so long, and he would like the issue quietly solved without alerting the Congress, the medical community, and the public. Implicit in the discussion was the concern that the FDA felt that this action was long overdue. He wanted a date, such as June the 1st, for the letters from us. Go over the page, please. Industry responded with a list of reasons why they had particular problems with the proposal, including the value of the inventory in our control, the lack of inhibitor indication for PTC, Alpha's pending dry heat approval, Highland's non-approval for Autoplex, etc. Everyone had a reason, and Highland mentioned an 18 million unit AHF inventory that would have to be reworked to heat-treat product. The industry position was that we would need rapid review of pending submissions and some time to review the situation with our management. Several proposed a staggered elimination of non-heat licence, starting with AHF and moving into other areas as feasible. The international situation was reviewed extensively. Mayer replied that he understood the situation and could sympathise with the difficulties, but that did not remove the overriding concern that no one, anywhere in the world, should be allowed continued exposure to HTLV-3 for any of the reasons mentioned. He specifically mentioned that the Japanese registration would soon occur, and he would assist with rapid review and approval for submissions, but the de-licensure of all products had to occur soon. I spoke with Aronson following the meeting, and he reported the problem with the review was that, quote, there are those who seek to license heat-treating processes with low temperatures and short times, which the FDA finds unacceptable. This was briefly reviewed during the meeting, and Highland, Dr. Holst, stated that Qatar had made it difficult for the rest of the industry by using such extreme temperatures for so long a duration, and thus established precedent. I could only respond that we had arrived at the time and temperature following extensive development work, and that our process was satisfactory for our product. I sensed that Highland is seeking to increase the severity of their inactivation step. The FDA is concerned about the limitations of both the alpha dry heat and New York blood inactivation processes. The FDA will meet internally during the interim to review the special situations. We agreed to review this internally and meet again with him on June the 17th to finalise our actions and the timetable. It is clear that Mayer intends to solve this problem quickly, with or without cooperation. That's where we will leave that document, sir.
Well, just if we take away the highlighting and go back to the top of that page, the last sentence of the first paragraph, everyone had a reason, and Highland mentioned an 18 million unit AHF inventory that would have to be reworked to heat-treat product. Is the implication from that Highland, at least, and possibly some of the others, had in stock non-heat-treated product which, despite the comparative lack of safety, as it was thought, they wanted to sell and intended to sell?
I think that is, well, it's certainly an inference that can be drawn, and perhaps might be the only inference from that sentence. I would also point to the reference to the international situation being reviewed extensively, and that is taken with Dr. Mayer's comments that nobody in the world should be exposed to HTLV-3.
Yes.
It seems to be the same issue that we looked at with the memoranda before is arising, that in countries which have not yet adopted a requirement for heat-treated products, non-heat-treated products might still be sold to them.
So the position was that so long as, or at least the implication is that the position was that so long as countries in the world which did not themselves take a regulatory position which prevented the use of non-heat-treated product could be sold and would accept non-heat-treated product.
That is certainly what I take from this document, yes, sir.
Yes. That would be a matter for them and their legislatures or organisations or their executive, whatever the particular situation in a particular country was.
And indeed for the doctors in those countries as to whether or not they, A, wanted to use that product, and B, had any choice in using that product.
Well, they might not have had clinical freedom in the same way as doctors in the UK did.
Indeed. They also may not have had access to the more expensive heat-treated products.
Yeah. Yes, thank you.
The situation at the end of that meeting is that there is going to be another meeting on the 17th of June. We don't have the minutes of that meeting, though we do have other documents which demonstrate that it took place. But even before the 17th of June, we see some of the fractionators preempting matters and taking their own steps. On the 3rd of June 1985, Highland announced that it was discontinuing production of non-heat treated haemophil and Proplex, and that it would recover any used units and replace them with heat treated products. The public statement commented that this was being done despite there being no clear scientific evidence that either of the products was involved in the transmission of AIDS. That's at paragraph 238. I note that Dr. Kingdon, in his 1990 statement, had said that Highland had stopped the sale of all non-heat treated factor VIII concentrates in the USA on the 31st of January 1985. And that might take us back to the point that you have just made, sir, that these were still being produced as of June 1985, presumably for sale elsewhere. Not necessarily Britain, I should add.
Yeah.
The inquiry also has a document from Dr. Riddell of Armour. He wrote on the 12th of July that Armour was no longer manufacturing or distributing untreated anti-haemophilic factor human for domestic utilisation. So no longer making or selling unheated products in the US. He said that the company was still continuing to export unheated products to Germany, but that was, and I quote, for heat treatment at that site. That was being done because that was the more efficient way of providing that product to markets that were supplied out of Germany. There is no reference in Dr. Riddell's letter to Armour withdrawing or replacing the non-heat treated products. And if we could have on screen, please, CGRA 40396. This is a Qatar document which tells us something of Qatar's response. If we could have page 2 of this, please. You can see it's entitled Coagulation Update, 28th of June 1985. What it says is this: non-heat action. Effective 21st of June 1985, all non-heat treated Co8 and Co9 were discontinued for sale in the United States. A letter was sent to all customers who purchased these products in 1984 and 1985, announcing action and recommending return of any existing product. Internationally, a telex and bulletin were sent to all distributors and all affiliates, announcing similar action. We are not requesting return of existing inventories or non-heat treated product internationally due to difficulties in customs, licenses, etc. No estimation of the volume of returns to Clayton available to date. The NHF and WHF, that's the National Haemophilia Foundation and World Foundation for Haemophilia, have complimented Qatar for the action. The phone calls received from customers all have been very positive, none have been negative. Most common remark is that of appreciation and admiration for the responsibility shown by Qatar. Additional comments have been on the completeness of the announcement package as compared with Highland's letter. If we go on, please. This is describing the situation of Qatar's competitors. This is Qatar's understanding of the situation. Competitively, Highland announced similar action on the 11th of June 1985, but did not remove non-heat treated Proplex and Autoplex. I pause there to say, so that's not entirely consistent with the letter that we have, and which I refer to at paragraph 238 of the written presentation, which does suggest that Proplex is being replaced. It may be that Highland's position on it changed, or it may be that Qatar have misunderstood the situation there. On the 19th of June 1980, returning to the document, on the 19th of June 1985, Highland recalled 4 lots of Proplex due to an AIDS donor in the plasma pool. Armour is reportedly not withdrawing any non-heat-treated product. Alpha has reportedly sold all existing non-heat-treated Profil 9 to the New York Blood Center, leaving them with no existing inventory and no plans to make an announcement. I think we can take that document off the screen now. Counsel to the inquiry haven't been able to identify any documents from Alpha itself setting out its position and actions at that time, but that Qatar note that we have just looked at would seem to imply that all of Alpha's Factor 8 product, Profil 8, was being heat treated at that time. So that may be why there is no specific announcement. It appears that these steps that were taken by the fractionators were sufficient to avoid the imposition of further regulatory requirements. In his
further,
and indeed any regulatory requirements in this.
Because the earlier March plasma rate, that wasn't a regulation; it was a recommendation.
It was a recommendation, yes, and even I think recommendation can sometimes have a technical meaning, and I don't think it even had that in the FDA. It was a public statement, as it were.
Yes.
So these steps avoid the imposition of any regulatory requirements. Dr. Kingdon said that he didn't recall any general product recall, and the Crever report recorded that it was not until 1989 that the FDA required the recall and destruction of all non-heat-treated factor products. In his tragic history, Dr. Everett wrote that by the beginning of 1985, little non-heat-treated factor 8 was being used anywhere. If we could bring up, please, CVHB 5042 on page 7 of it. If we could go down to the section just above acknowledgements and expand the final paragraph. There's a short explanation of some of the meetings that we have discussed earlier. What Dr. Everett goes on to say is this, and I quote from about halfway through that final paragraph: "The world's haemophilia community quickly adopted the MASAC, that's the National Haemophilia Foundation, recommendation, so that by the beginning of 1985, little non-heated clotting factor 8 was used anywhere. The AIDS epidemic in the haemophilic patients thus suddenly ceased. Subsequent studies of birth cohorts demonstrated that no haemophilic patients born in the USA in 1985 and later were identified with LAV."
Infected.
I'm sorry, sir?
No haemophilic patients born in the USA in 1985 and later were infected.
Were infected with LAV, later to be renamed HIV. So 1985 onwards, no person with haemophilia in the United States was found to be infected if they were born in 1985 or afterwards. Tragically, during the period 1981 to 1984, more than 50% of the population of haemophilic patients in the United States had already become infected. And these individuals would continue to present clinical symptoms of AIDS during the next decade. and many would die. It's on that note that Dr. Evatt concludes his tragic history. I would add to that while the epidemic of AIDS cases may have been brought to an end by the use of heat-treated products, there were still a number of seroconversions that were associated with the use of heat-treated products. In a review article from 1995, which we looked at in September and October, Professor Pierre Minucci referred to 18 well-documented cases of HIV transmission through concentrates that were subject to dry heat treatment. Many of these were patients who used batches of factor VIII HT that had been subjected to heat treatment, but which contained plasma that had been obtained before the HTLV3 screening test was introduced. ARMA withdrew such products from the UK market following consultation with the DHSS in September 1986. My understanding is that those products weren't withdrawn from other markets. The inquiry will hear further evidence on this matter, and I won't go into it in any further detail now. It's also important to note that while heat treatment, and particularly heat treatment in combination with the HIV screening test, was effective in inactivating HIV in these products, the first generation of heat-treated products did not, in general, prevent hepatitis infections. By the mid-1980s, most such infections were of non-A, non-B hepatitis, but there were still cases in which heat-treated products were implicated in infections of hepatitis B. The evidence of Dr. Vivian Mitchell from the Leicester Haemophilia Centre and a letter that he wrote to the Lancet in the 1980s are evidence of such infections. You heard about those at an earlier stage in the inquiry. That is where I will leave the story of heat treatment. But of course, the inquiry will be hearing further evidence from domestic fractionators and about domestic fractionators, and heat treatment plays an important part in that story as well, of course. I should also flag that at the end of this presentation, I will also briefly speak about what's often referred to as the Prince controversy, about Dr. Alfred Prince and his work on ARMA's heat-treated product, and I'll come on to that a little later. But for now, I'd like to turn to the screening test for HTLV3. I stress that this part of the presentation is about the situation in the United States. The inquiry will hear further evidence about the United Kingdom position in due course. We discussed earlier, sir, Secretary Heckler and her announcement of the isolation of HTLV3 on the 23rd of April 1984, and her prediction that a test would be available within 6 months. In fact, that was to prove optimistic. In June of 1984, the United States authorities announced that 5 firms had been invited to develop and distribute HTLV3 antibody tests. They were doing so in cooperation with the US federal authorities who were providing them with the assays. The first application for a license for a test was made in December 1984. The fractionation companies, of course, were aware that this was going on, and in September 1984, a conference call took place between representatives of the main companies, at which a common position was sought on issues including informed consent, the information to be provided to rejected donors, and the ever-contentious matter of the recall of previously fractionated products where plasma from a donor had tested to be HTLV3 positive. Dr. Hink provided a memorandum of recall, which he succinctly summed up by saying, quotes, "There was a lot of talk, which led to nothing conclusive." So that's September 1984. But by the 15th of February 1985, an industry position had emerged, and we can see that position being set out by Robert Riley of the American Blood Resources Association in a letter to Dr. Patriciani. If we could have on screen, please, BAYP 305650. If we go down to the third paragraph, please, starting with the advent. What Dr. Riley wrote is this: with the advent of anti-HDLV3 testing, the manufacturers of blood derivative products are faced with a dilemma similar to that which arose with HBsAg, hepatitis B surface antigen testing, i.e., 1. What steps should be taken when a donor who was given plasma in the past is determined to be positive to a test for anti-HDLV3? 2. Further, what should be done with such a donor's plasma still stored at the plasma centre and/or at the plasma product manufacturer's location? 3. What should be the disposition of in-process plasma products containing such units of plasma? There are several facts that have emerged which are pertinent to a discussion of the above. First, it is estimated that approximately 0.05% to 1.0% of the normal donors will react positively to the anti-HDLV3 test. Second, at the very minimum, pools of plasma used in plasma fractionation include at least 1,000 donations of plasma, which would mean that it is reasonable to assume that currently all lots of product produced contain plasma with anti-HDLV3. Third, the risk of transmitting the AIDS virus through plasma products is nearly non-existent, with the exception of non-heat-treated coagulation products. Go over the page. Therefore, our proposal for the handling of the items listed above is as follows. 1. Intact units of plasma collected from donors subsequently found to be anti-HDLV3 reactive in storage either at the plasma centre or the manufacturer's location and which are not yet in process would be segregated and disposed of, as would any other plasma found to be anti-HDLV3 reactive, i.e. destroyed or used for other purposes as may be approved by the FDA. 2. For in-process products containing anti-HDLV3 reactive plasma, no action should be taken. The basis for this proposal is that it becomes obvious that if we were faced with the alternative, that is, removal of these lots of products from the market, there would be either a total lack of availability of these life-saving products or, at the very least, a very acute shortage of them. There is a second dilemma still currently facing fractionation manufacturers, and that is the disposition of lots of coagulation factors manufactured using a heat treatment process which has been shown to inactivate known amounts of viral contaminants and which has been made from a plasma pool which contains a donation or donations from a confirmed AIDS patient. In this case, we propose that the manufacturer should take no action if he has data showing inactivation of the HDLV3 virus by this heat treatment process. That is the position of the fractionation companies put forward. And if we just go down, thank you, we can see that the letter is copied to Penny Carr at Alpha, Steve Holst at Highland, Steve Ajala at Qatar, and Michael Riddell at Revlon but Armour. So proposal seems to be that if there is a unit of plasma which is found to be positive for HIV antibody and which hasn't been used in any products and isn't being processed, then it will be destroyed. But if the HIV positive donor is found and the plasma from that donor is already being processed or is already in a product, so long as that product is heat treated, there is no need to withdraw or recall or prevent it being distributed. That is the proposition that the companies were putting forward. That's on the 15th of February 1985. On the 22nd of February 1985, a public meeting took place on this issue. Forgive me, on the 22nd of February, it wasn't specifically on this issue. It was a public meeting about HTLV-3 screening. There is a memorandum, which I summarise at paragraph 254 of the written presentation. That comes from Dr. Ajala. He refers to this as being, and I quote him, quotes, almost a press conference rather than a scientific meeting, and I believe this approach was intentional, close quotes. And he goes on to explain that what he meant by that was that he thought that this was the FDA and the CDC using a public meeting to show their confidence in the HTLV-3 screening tests that they had helped to produce. And Dr. Ajala said that that was necessary because of the negativity that had been shown towards the test by a number of representatives of the blood banking industry and what he refers to as special interest groups, which is presumably a reference to gay rights organisations. So there is a test which has been produced by the FDA and the CDC and the companies involved. There is hostility to that test from the blood bankers and from gay rights organisations. But according to Dr. Ajala, Bob Riley spoke for the plasma industry. Begin quote, he did not say very much specific other than we thought prompt approval of a test was a good idea, close quote. So again, we see a distinction between the approach of the fractionators and the blood banking industry. At the request of the fractionators, a meeting took place with the FDA on the 26th of February 1994. And that was to discuss the specific issues that have been raised in Bob Riley's letter, the question of product recall. It was preceded by an industry-only meeting, as was common, and according to a Qatar memorandum, there was, and I quote, no enthusiasm or desire, end quote, among their competitors to contest the HTLV screening test. They did say, however, that they didn't want industry to be burdened, as it was put, by the confirmatory test, the Western blot test, which would be used to confirm that a donor was HIV positive. The quotation contained in the memorandum indicates that industry would simply tell a donor that they were being excluded. And so far as they were concerned, that was the end of the matter for them. And the quotation is, and I quote, industry cannot afford to deal with running confirmation tests. So presumably donors would be left to go and seek confirmation on their own. The meeting also discussed the imposition of ALT testing for the German market. That is something that I'm going to come back to in a second. We can see from the memorandum that Dr. Riddell suggested that the industry might want to put forward a proposal to the FDA about ALT testing in order to avoid regulatory action being taken. We'll come on to see what that discussion led to. The reference to the memorandum to which I'm taking this from is BAYP 305664. That's a reference which I've omitted from the written presentation and will seek to rectify that. So the meeting took place with the FDA on the 26th of February 1984. I won't take you to any further note of it because, in essence, the fractionators set out the position as per Dr. Riley's letter, and Dr. Patriciani listened but didn't commit the FDA to any response on it. Following that arranged meeting, an informal meeting took place, and I'm going to take you to this, please. It's CGRA 40312. This is a memorandum written by Dr. Carr of Alpha. The memorandum is dated the 1st of March 1985, but it refers to the meeting with the FDA on the 26th of February 1985. Reading from the top, this is what Dr. Carr wrote. I had requested a meeting with Dr. Patriciani after the fractionation industry meeting to include only the 4 commercial manufacturers. Present were Bob Riley, that's of the trade body, Mike Riddell, Armour, Steve Holst, Highland, and myself. Steve Ajala, Qatar, had to catch a plane. We had all agreed that we would introduce the subject by asking the status of the study we had, I should have said that the subject heading is meeting with FDA on prison source plasma. We had all agreed that we would introduce the subject by asking the status of a study we had heard the FDA was performing on prison plasma, and Bob Riley did so. Dr. Patriciani went briefly into the background of why they decided to institute such testing. He became aware that they were receiving more applications for source plasma establishments in prisons. They had had problems in the past with the shipment of hepatitis B surface antigen reactive units from prison sources and falsification of records. With the concern over AIDS and concern over the conduct of any kind of studies or activities associated with prisoners, they felt that if there were more and more prison sources of plasma, that they were in a very uncomfortable position over how FDA could justify these with no scientific data, particularly in the socio-political atmosphere. If challenged, he believes they must have a scientific basis for allowing this to continue. Otherwise, this could be a political bombshell. I pause there, sir, to note, this brings us back to the discussion we were having before, that there is a continuation of plasmapheresis stations within prisons in the United States in this period. It may be that the plasma being obtained is not being used in coagulation products, but it is still being obtained, and the FDA are now undertaking a study to see whether or not there is a difference between prison plasma and other plasma.
The implication, as I see it from the second paragraph there, is that there's not only a continuation and there's an expansion.
Yes.
Because that's the force of the word more.
Yes. Yes.
So whoever is collecting the plasma is marketing it somewhere or
Yes.
using it for some manufacturing purpose itself.
Yes. The memorandum from Dr. Carvin goes on to describe the parameters of the study and the way that the FDA are approaching it. There's a short paragraph, 3 paragraphs down, which says, and I quote, if prison plasma can't be differentiated from non-prison plasma, they would then consider relaxing the guidelines. It's not entirely clear what the guidelines referred to are there, but it may be an inference that that is a reference to the guidelines from March 1983 about the way in which fractionators should avoid high-risk areas.
Yes, but the high-risk areas there didn't include prisons, did they?
I don't know if prisons were specifically mentioned, but there is what is referred to in this and other documents is a gentleman's agreement that prison plasma wasn't going to be used. And so it may be that the reference to guidelines is intended to capture that agreement. But there is certainly no regulatory action which has been taken about prison plasma. You'll recall from the letter of the memorandum from Highland, there was a conversation with Dr. Donoghue about what Highland's position was going to be. And Dr. Donoghue was offered a written statement from Highland, and he said that was not something that he would say.
Yes.
The memorandum then goes on to talk about the potential target date for the studies. And then at the bottom of that page, if we pick it up again, and I quote, when asked to what their reaction would be if a manufacturer wanted to do such a study, he stated that they would consider it good complementary information. Turning over the page. I informed him that since we had made these arrangements 2 years ago...
Agreements.
I informed him that since we had made these agreements 2 years ago, much more had been learned. And with a shortage of plasma now and more expected with the advent of HDLV3 testing, industry management was questioning why the differentiation from other donor sources. I stated this for his information, simply to prepare the way for any further inquiries we wish to make on our own. Note that Armour apparently has absolutely no interest in this subject, but was willing to be present. Steve Holst obviously believes Highland does have an interest, but was really non-participatory since he is so new to his job. Although Bob Riley was willing to participate in discussion on this, he obviously considers this, as stated, a potential bombshell and does not want to press further since it does not represent the majority of his constituency. His constituency, I should add, is the American Blood Resources Association, so plasma manufacturers and plasmapheresis centres. Returning to the document, quotes, if we wish to press further, it would be my recommendation that we try doing so with Highland and Qatar, end quotes. And if we look at the marginalia on the document, written is the question, do we want to pursue? And in answer to that question, sent to Pete DeHart and signed by Vaughan, it says, Pete, how much material do we get currently? I have my
Brackets, prison plasma.
Prison plasma, thank you. My written note is somewhere else. Could this represent an opportunity to
source substantial increased amounts?
Thank you. If yes, we probably do want to pursue. The reference there to getting currently might suggest that Alpha is one of the companies which is obtaining plasma from prisons, albeit they've entered into this gentleman's agreement not to fractionate it.
Well, an agreement, let's call it.
An agreement.
Whether it was between gentlemen, we wait to see the rest of the evidence.
There were quotation marks there because it's a phrase which is used in one of the other documents. So that is a memorandum from the 1st of March 1985, referring back to that meeting on the 26th of February, which was about both HDLV testing and then, as a consequence of HDLV testing and heat treatment, the possibility of revisiting the issue of prison plasma with the FDA. On the 2nd of March 1985, the Department of Health and Human Services announced that the first HDLV3 antibody test kit, which had been manufactured by Abbott, had been licensed by the FDA. In his 1990 draft witness statement, Dr. Kingdon stated his opinion that the 7 months that it took between the approval of the companies to begin working on this project and the licensing of this test kit was, and I quote him, in scientific terms, a remarkably short period. Dr. Ajala, in a memorandum, refers to the CDC and the FDA considering that they have made, and I quote, Herculean efforts in order to get the test licensed so quickly. Armour and Highland began testing donors that month. However, according to Dr. Kingdon's 1990 draft statement, there were insufficient test kits available to satisfy US demand until June of 1985. The Creever report says that HTLV-3 testing became available in all centres by July 1985, and it had been available in the vast majority of centres by May 1985. So just to give those dates in order again, March 1985, the FDA licence is granted. By May 1985, according to Creever, the testing is being done in the vast majority of centres, and in either June or July 1985, there is full capacity for the US market. Point to note now and come back to in the future is that Dr. Kingdon expressed his view in his draft witness statement that, and I quote, until there were sufficient kits available to meet US demand, it is unlikely that any of the test kit manufacturers would have been willing to export their tests abroad. End quotes. I leave that there. It's no doubt a matter to which we will return in due course.
The tests were developed before being licensed, presumably.
Yes.
Do we know when they were actually developed and therefore being in use, at least for a testing point of view in other jurisdictions?
I don't know that on the top of my head, but we can look into that. From memory, there is a helpful table in the Institute of Medicine report, I think, which provides a contrast between different jurisdictions and who was doing what when. There is always a difficulty in comparing like with like in tabular form, but that's something that I can have a look at in the break, and then we can also do further work on trying to understand.
And from what you showed me earlier, there was an LAV test, and LAV, it was exactly my understanding is, exactly the same basic virus that Montagnier identified, as did Gallo.
Yes.
So that was available in August.
August 1984, yes. The CDC had worked to develop that with the Paris team. They were using it, presenting results from it in August in order to do their laboratory work on viral inactivation through heat treatment. And in the UK, we also have the development of some form of testing in 1984.
Yes,
I won't try and remember the dates off the top of my head.
That I think may also be August at the earliest.
Yes.
But we may well hear from Dr. Tedderer in due course.
So these are tests which can be done on individual samples.
Of course.
What
yes. Plainly, to be used as screening tests, there has to be sufficient quantity to enable the screening to be done.
Just so, and the licensing of the test is the licensing not just of a final test, but of a process that leads to it. So it has to be a process at industrial scale in order to be able to roll the tests out across the country. That is what was licensed in March 1985.
Yes.
On the 12th of April 1985, there is a fractionators meeting ahead of a blood products advisory panel meeting, so another one of these industry-only meetings. If we could have on screen, please, CGRA 40261. This meeting sees the debate moving on to a related topic, which is the introduction of ALT testing. So that's a form of liver function test. That was to be instigated in response to the requirements of the German regulators that any plasma with ALT levels at more than twice the normal level should be excluded from plasma pools for factor products. The German regulator put that requirement in place. The American firms who wished to sell in Germany therefore had to adhere to it if they were going to be able to license their products there. It's apparent from the discussion that it was estimated that around 20% of US plasma would be tested in this way. So it's about 20% of US plasma would be used in products that were available for the German market. The issues for the fractionators were first, what was to be done with plasma that was, to use a word in quotation marks, positive for being twice the normal ALT level. That plasma couldn't be used for a German product, so what do you do with it? Second, how best to avoid the unwelcome, as the fractionators saw it, introduction of ALT testing for all of US plasma. With that introduction, we look at the document, which is again another memorandum from Dr. Rajala of Qatar, and an internal Qatar memorandum reporting on the meeting. The first paragraph, there's a list of attendees, and then the first paragraph says, Penny Carr, Alpha, Steve Holst, Highland, and I met in the Osaka room at Alpha headquarters from 9 to 12 on April the 12th, 1985. We followed this meeting with a 2-hour conference call to Bob Riley and Mike Riddell to confirm our plans for testing and the upcoming Blood and Blood Products Advisory Panel meeting. The following are salient conclusions. ALT testing. The Ajala script for an industry position was accepted with enthusiasm from Alpha and some reluctance from the others. Geraghty, that's a reference to Dr. Geraghty of the FDA, Geraghty or someone will express concern to the advisory panel that use of ALT-positive plasma in domestic products will change the character of those products. Our formal presentation will state that we are simply hand-selecting, via ALT screening, special plasma for a particular customer, German market, and that all remaining plasma remains licensed source plasma which may be utilised in other products. The ALT test is not required by the FDA, nor do we want it to become so. We will state that any plasma 2 times normal will not be used for the particular customer, that's the German market. We will further offer that any plasma with 5 times the upper limit of normal will not be used in domestic product, and the donor will be deferred until the ALT levels fall back to normal. We will use the argument that 5 times normal may indicate a donor health problem. This may not please all of you, but it was the only way to achieve industry consensus. Further, plasma in the over 2 but less than 5 range will be used randomly in production so as not to concentrate this plasma in any particular lots. All coagulation products will be heat treated for an extra margin of safety. We will endeavour to call German plasma special, or some other term, to nullify the idea that greater than 2 times normal plasma is, quotations, positive or other negative connotations. In practice, Armour will discard all 2 times cryo, and Highland will discard all 2 times plasma from coagulation products and fraction 2 products. We estimate that approximately 20% of total US plasma will be screened, but will vary between manufacturers. ARMA requirements are modest, and Highland will screen all plasma for the interim to, quotes, fill the pipeline. Alpha will screen only A and B plasma from select centres. Highland's conservative position is due to ARMA's decision, i.e., they want to be perceived as conservative in any subsequent litigation. Because they had 2 patients develop non-A, non-B in a clinical trial of their intravenous immunoglobulin in Seattle. Holst thinks it's because of procedures at the clinical site. That's a reference to why he thinks there are positive results there. But Penny thinks it is because of starting with 2 + 3 paste for the Highland product. The FDA has summoned Highland to Washington for a summit conference on the Seattle incident. This is delaying their intravenous immunoglobulin approval until resolved. In summary, the minimum industry position is 1, less than 2 times normal, German product. 2, 2 times normal to 5 times normal, random distribution in domestic product. 3, greater than 5 times normal, discard plasma, defer donor. This hasn't been approved by FDA, and Highland and ARMA will react more conservatively. I suggest this item be reviewed at the next BMC. Bob, that's at the internal Qatar committee. Bob Riley also mentioned that we will need to make some provision in our business arrangements with our contract centres to preclude them from dumping high ALT plasma on us. Prison plasma. This subject elicits even more diverse viewpoints. Qatar and Alpha believe that science has progressed to the point that we can screen this plasma through testing, HTLV3, etc., and we now heat treat the products. Highland says they have no current prison plasma sources. An exclamation mark is contained in parentheses. And ARMA states they will never have any. Riley is perpetually gloomy on the entire subject and feels we are destined to fail. Nevertheless, we agreed to hang together for a try with the FDA. We will propose to begin using Prism Plasma Cryo and abandon our gentleman's agreement unless the FDA takes issue and threatens regulatory action. We will further agree to do whatever testing the FDA deems necessary to answer any academic concerns. Sam Anderson will contact the NHF to ensure there are no major obstacles there, and I recommend Jack Ryan does likewise. It will not serve our purposes to effect change with the FDA and offend our customers. The argument for using Prism Plasma is the additional testing, HDLV3, and heat treatment, which provides product safety. Penny, Mike, and I will try to discuss this with Patriciani in Atlanta this week on a preliminary basis. If we could go over to the next page, please. This one final extract from this minute. FDA letter. No one likes the FDA response to our meeting in February to review the position on recall. We will lobby for a change to 3 or 6 months for unpooled units, not 3 years. But in any event, the letter has not been officially sent to the manufacturers. New York Blood is doing retrospective testing on all plasma samples, and we agreed it was a bad idea. They will inform transfusion victims of the results. There is a little bit to unpack there, so 3 distinct topics. On the ALT testing, our understanding of the idea being put forward is that 20% of US plasma, approximately, was going to be tested for ALT. where plasma was found to have ALT levels that are less than twice the normal limit, however normal was defined, that plasma would be used in products that would be sold to the German market. Where plasma was above twice the normal limit, but below 5 times the normal limit, that plasma would then be placed in a general pool with the untested plasma, and that would be used to make products that would be available for the American market and presumably other international markets that didn't have the same regulatory requirements as the German market. And that is why it's a reference to it being randomly distributed across the plasma pool, rather than that plasma alone being used to make products which would inevitably have a higher ALT level.
Can you just help me with this? The assumption may well be that the German requirement is imposed to protect German consumers of blood products from the risk that high ALT level blood is infected with a virus which will transmit hepatitis.
Yes.
So it's quite possible, if they're right, that that blood has a hepatitis virus in it. If blood with hepatitis virus in it is mixed in a pool with other donations, it was the medical understanding since the early 50s that it would take only 1 or 2 donations to infect the whole pool.
That is my understanding of the risk assessment.
So the proposal to mix high ALT blood through all the pools would render all the pools potentially more hazardous than they might have been.
Potentially, yes.
Yes. Thank you.
The plasma that was more than 5 times the normal limit would be discarded, but the reason for that being discarded would be given as donor health. So not a suggestion that the plasma was unsafe, but a concern for the donor. The other point that I should make about that is that the German requirement for ALT testing may well have been about hepatitis. There were also some suggestions that ALT testing could be a surrogate marker for HIV. I'm afraid I don't know the reason why the German authorities put in place this request, whether it was hepatitis or AIDS or both.
Well, you may want to check this, but I think it may be in his judgment in the A and the NBA case that Mr. Justice Burton may have identified that in Germany, ALT testing of blood for transfusion, so not the same as plasma for plasma products, was introduced in the 70s. But that may need to be checked.
I will have a look at that, and again, that may be possible to see that over the break. Otherwise, we can return to this topic.
Yeah, well, in due course, it's not a matter of immediate importance, but it's something which I thought I'd better mention it because it's something which I recollect from memory. I may be wrong, but you'll want to check it out.
I will do so. What I would add, though, is that even if a stated reason of AIDS was given for the test, ALT is about liver function.
Yes.
Inevitably, hepatitis is a relevant consideration when testing for ALT levels. So that's the ALT testing part of the discussion. The prison plasma discussion leads to a suggestion that despite some disparate views within the group, they are willing all to put forward a proposal to the FDA to see what the FDA will say in response to a proposal of going back on the supposed gentleman's agreement on the use of PRISM plasma. And there are suggestions of making contact with the National Haemophilia Foundation, and we'll come back to that in a second. The third issue is the FDA's response to the letter that had been sent in February and the meeting that had taken place in February about product recall. And I'm afraid our documentation, or at least the documentation we have identified, is limited on this. So we have a rather opaque minute, and I'm wary about trying to decipher that. It appears that the FDA have come up with a counter proposal, which involves some kind of period of look back or of quarantine for products. And the industry is going to lobby for a change to 3 or 6 months. It says not 3 years. I don't know if that is the period that the FDA had proposed or whether or not that had come from somewhere else. But I'm afraid, because of the limitations of documentation, I can't assist much further on that point. What we do know is that Jack Ryan, who was referred to in this minute, did make contact about PRISM plasma with Dr. Charles Carman and Dr. Peter Levine of the National Haemophilia Foundation. On the 1st of May 1985, there was a conversation between the three men. From a Qatar document, it's recorded that Dr. Levine responded to the proposal on a personal basis, that he thought reflected the views of the NHF. And what he said was, and I quote, "He still feels that we are taking risks that are not justified, specifically increased risks for hepatitis non-A and non-B, as well as hepatitis B," close quotes. Just referring back to the discussion that we have looked at, there is the justification for returning to PRISM plasma is framed in respect of both heat treatment and the introduction of HDLV3 testing. Now, heat treatment had been demonstrated by that period to be effective in respect of HIV, and HDLV3 testing was, of course, concerned with AIDS, but there is still a hepatitis risk, which, as we know now, had not been addressed by either of those measures, or not, certainly not fully addressed. Hepatitis viruses were not inactivated by most first-generation heat-treated products. Jack Ryan of Qatar informed Dr. Levine and Dr. Carman that Qatar would continue their dialogue with the FDA, as they thought that the company's position was, and I quote, "safe and justified." Counsel to the inquiry haven't identified any further documents in which the use of PRISM plasma was raised, and we are unable to comment on what, if any, changes in practice were made. And similarly, it's not clear from the documents seen by Counsel to the inquiry what decisions were reached on ALT testing, although it does seem that it became more prevalent in the years that follow. I'm afraid the presentation rather ends here, because it is a presentation primarily about the response to AIDS, and some of these issues obviously continue into the 80s. It's perhaps worth referring back to a document that we looked at in September from Dr. Carl E. Brooke, who was the president of Highland, who sent a letter to the National Haemophilia Foundation in 1987. You may recall, sir, it's a letter which sticks in the memory, with Dr. Brooke criticising the National Haemophilia Foundation for seeking low-cost product, which, on his argument, had pushed for fractionators to look for low-cost sources of plasma. In that letter, Dr. Brooke, defending Highland's position, stated that the company had been, and I quote, the first manufacturer to screen all plasma in compliance with German BDA requirements in 1985. I pause there to note that this is the conservative position, as it was referred to by Dr. Rajala, now being used a couple of years later in support of Highland's position. He also claimed that Highland had behaved in a responsible fashion by not using material rejected for Germany's ALT requirements in any other market, as several other US manufacturers did. And so that was Dr. Brooks' comments. As I say, so we have reached the end of the chronological period covered by this presentation. And just to summarise the position as of July 1985, all US fractionators by that time were producing heat-treated factor concentrates from plasma screened for HDLV antibodies, HDLV3 antibodies. Donors were subjected to screening regimes that were intended to exclude high-risk donations, including from gay donors. This led to a high level of protection against transmission of HIV, but not, in most cases for these first-generation heat-treated products, against non-A, non-B hepatitis. The protection against AIDS afforded by these measures had led to the reversal of the previous approach to withdrawing batches of factor concentrate that were known to have been contributed to by donors who were later diagnosed with AIDS. So such products were not being recalled if the product had been heat-treated. Some fractionators were considering going further and had opened a dialogue with the FDA with a view to reintroducing the fractionation of plasma from prison donors in coagulation products. This was an issue that had split the pharmaceutical companies, not least because of the anticipated increased risk of hepatitis. But as we have seen from that last memorandum from Dr. Rajala, even those companies that were not proposing to do this themselves were willing to, as Dr. Rajala put it, hang together in an approach to the FDA. The introduction of ALT testing in respect of plasma destined for the German market had posed a new set of problems for the fractionators. A consensus had been put forward for a proposal that was intended to ward off mandatory testing of all US plasma for ALT levels. That proposal, if it were to be implemented, would have meant that products exported to Germany would be produced from plasma specifically selected as being relatively low in a marker for hepatitis, whereas domestic products would be drawn from plasma that either had not been tested or was known to have higher ALT levels. That is where the main part of the presentation ends. There is a short appendix about Dr. Prince, and I note the time. I don't know if that is something which I imagine would take about 20 minutes.
Well, shall we go ahead and deal with it?
Very well, sir. In December 1984, so we've gone back in time a little, December 1984, ARMA commissioned Dr. Alfred Price of the New York Blood Center to undertake studies on the efficacy of its heat treatment process for factor VIII. The purpose was to see whether a measured quantity of HDLV3 would be inactivated by the heat processes that ARMA were using. The proposed approach was to take a known amount of the virus and add it to the concentrate, so to spike the concentrate, before the concentrate was freeze-dried or lyophilized, and before it was then heated. Following that process, the product would be tested to see how much residual virus it contained. Virus reduction was measured by determining how many logs of inactivation had been achieved. So if a sample in which the amount of virus is reduced in size by 10 to the power of 4 millilitres is achieved, then it is said that there has been a 4-log reduction, or a 4-log kill. The industry standard at the time was a 5-log reduction. It was anticipated that virus could be inactivated both by the freeze-drying process itself and by the subsequent heating of the product. Armour's heating technique involved dry heat at 60 degrees centigrade for 30 hours. We looked at this earlier in the table. Highland's equivalent product was dry heat 60 degrees for 72 to 74 hours. Qatar's product was dry heat 68 degrees for 72 hours. Just as a point of comparison, the heat-treated product produced by BPL in the UK, 8Y, was heated at 80 degrees centigrade for 72 hours. I note in parenthesis here, sir, that although figures of 60 degrees and 10 hours were given for some of the other products, those were products which were not dry heated, but were heated either in solution or in suspension and so forth. So Armour, 60 degrees centigrade for 30 hours, is a less robust heat treatment than its rival dry heat competitors at the time. Dr. Prince provided the results of his first study in January 1985. He wrote, and I quote, "Disappointingly, we were unable to show the greater than 5-log kill as had been hoped. The most that can be concluded from the study is that the combined effect of lyophilization and heating inactivated more than or equal to 2.5 to 3 logs." Dr. Prince's results were at odds with those that had previously been obtained by experiments conducted by the Centres for Disease Control. On the 29th of November 1984, Dr. Evett had written to Armour with the results of those studies, and Dr. Evett said that they indicated that heat treatment at 60 degrees for 24 hours, so less time than Armour used, had left no detectable virus in a sample of factor VIII concentrate. And his letter concluded, and I quote, "Because LAV appeared to be extremely heat labile, we believe that the procedures presently used by the manufacturers for heat treatment of hepatitis virus would adequately inactivate the LAV virus." That's Dr. Evett's view, as expressed in late November. The CDC results were subsequently published in the Journal of Clinical Investigation in August 1985. The lead authors were Dr. Evett and Dr. McDougall, who we heard about earlier. In October 1985, they had joined with Dr. John Patriciani of the FDA in writing a letter to the Lancet, which advocated heat treatment on the basis that heating at 60 degrees Celsius for 20 hours would result in a 20-log reduction.
A 20-log?
20-log reduction. Now, Dr. Prince would later criticise the assumptions that lay behind that article. But it was an article that was published in The Lancet in October 1985. Going back in time to Dr. Prince's initial studies, that's January 1985. So at this stage, Armour have been informed of the CDC's experiments in November 1984, but it's obviously before that Lancet article is published later in the year. At that point, January 1985, ARMA was left with conflicting studies concerning the safety of their heat treatment process. In an internal company memorandum on the 11th of February 1985, Dr. Riddell identified for his colleagues at ARMA limitations in Dr. Prince's study. In particular, he identified the low quantity of active virus that was included in the sample that was used by Dr. Prince. Dr. Riddell considered that the experiment should be considered preliminary in nature. He said that Dr. Prince's team had been provided with samples with higher level of virus, which would be used in further experiments in the coming weeks. The company would also go on to commission further studies from other respected sources. Dr. Prince and his team did indeed conduct the further studies, and they reported them to ARMA during the course of 1985. His findings were that heating at 60 degrees in the dry state had only, and I quote, a modest process efficacy for inactivation of HTLV3/LAV, end quote. This, he said, was a finding that was in contrast to the results obtained by Dr. McDougall of the CDC, and he said that this was, and I quote, difficult to explain. The implication of Dr. Prince's results, though, was that ARMA's established heat treatment process may not be sufficiently robust to inactivate HTLV3 to the industry standard of 5 logs. Dr. Prince wanted to publish his results, and he sent a draft article to ARMA. The company invoked a clause in their contract that required him to obtain ARMA's permission for any publication. That permission was not forthcoming. We could have on screen, please, CGRA 40512. This is a memorandum from the 8th of November 1985, and it is from Dr. William Terry of ARMA, and it is noted to be for the record. If we could expand the main text, please. Through the efforts of Bernie Loeb, we were able to determine that there is a contractual arrangement with Dr. Prince which precludes him from publishing, without our approval, data from experiments supported by RHCG, that's Revlon Healthcare Group, the parent company of ARMA.
Yep.
Dr. Prince submitted a manuscript to me entitled Safety of Blood Derivatives Pasteurized in the Dry State. The data in this manuscript indicate that Generation 1, Generation 2, and Factor 9 are not rendered virus-free when they have been contaminated with HTLV3 and heated in the dry state at 60 degrees for even as much as 72 hours. I pause there to note that Generation 1 and Generation 2 are different types of Factor VIII. I told Dr. Prince that while our foremost concern was the safety of patients receiving these products, these data, taken in isolation, could only be confusing to the scientific community, the treatment community, and the public, and that we were, therefore, not prepared to give him permission to publish. I pointed out the discrepancies between both his test system and his test system results when compared with the data reported by McDougall and others. I indicated that we certainly wished to continue working with him and hoped that we could do a more comprehensive study utilizing his sensitive test system, but applying it to the heating conditions and the products of all the major suppliers. While Dr. Prince was obviously disappointed and somewhat contentious, he accepted the fact he would not be able to publish these data, and the conversation ended on a reasonably cordial note. In later litigation, Dr. Prince said that he had been annoyed with Armour and that he set out to repeat his experiments outside of his contract with the company so that he could publish the results. He did so in a letter to The Lancet on the 31st of May, 1986. In that letter, he said that he found viral inactivation from heating at 60 degrees was on its own, and I quote, surprisingly modest. He identified viral inactivation of between 0 and 1 log when the sample was heated at 60 degrees for 10 hours, and between 2 and 4 logs observed after 72 hours of heating. The freeze-drying process on its own resulted in an additional 0.5 to 1 log of inactivation. If we could have PRSE 302534, please. This is the letter to The Lancet, effect of heat treatment of lyophilized blood derivatives on infectivity of human immunodeficiency. And if we could turn to the second page, please. We could just expand the final paragraph of the left-hand column, beginning the finding. Dr. Prince wrote this: the finding of only modest sterilization process of efficacy for HIV adds to concern about the efficacy of this procedure. It should, however, be stressed that this finding does not mean that dry heat-treated products are unsafe with respect to transmission of AIDS. Indeed, 3 studies have reported absence of anti-HIV seroconversion in recipients of dry-heated factor VIII preparations. Purification and processing steps before lyophilization can remove or inactivate virus, and lyophilization alone under commercial conditions probably inactivates more virus than is observed with shell freezing. Furthermore, some products are heated above 60 degrees Celsius. Nevertheless, these findings indicate the need for caution in relying on the efficacy of dry heat sterilization. Long-term surveillance of recipients of such products for seroconversions to anti-HIV is still required. If we could take that down, please. The approach taken by Armour to Dr. Prince's work has been the source of intense controversy for decades, not least because, as we've heard, heat-treated factor VIII came to be associated with a number of seroconversions. Dr. Peter Jones gave evidence to the Lindsay inquiry in Ireland about...
Can I just mention, for the sake of the transcript, that they've rendered your factor VIII as factor V111, whereas what you meant it to be, I think, was the name of the product itself.
That's
factor
right.
VIII.
Yes.
ATE.
Factor VIII ATE heat-treated. The Armour product was associated, as we've heard, with 18 seroconversions by Professor Minucci.
And I mentioned that so that when the transcript is perfected, that can be picked up.
I'm grateful, sir. Thank you. Peter Jones gave evidence to the Lindsay inquiry about a particularly contentious meeting of Armour executives and scientists that was held in October 1985. And in Dr. Jones's opinion, and I quote, the record of that meeting, quotes, the company appeared to consider it ethical to continue to market unsafe product whilst attempting to manipulate the results of their laboratory tests in order to throw doubt on the safety of their competitors' products in the eyes of the FDA. They are doing this in order to protect their share of the market. That was evidence given by Peter Jones to the Lindsay inquiry. Mr. Justice Creever came to the following conclusion in his report. Could we have, please, KREV 601, electronic page 529. It's the final paragraph above the subheading, beginning not only. The reference to the Bureau here is to the Canadian regulator, the Bureau of Biologics. And I quote, "Not only did ARMA not transmit Dr. Prince's data to the Bureau, but it prohibited Dr. Prince from publishing his research on the safety of its products. ARMA reasoned that the publication of Dr. Prince's research in isolation, and this is a quote, in isolation could only be confusing to the scientific community, the treatment community, and the public," end quote. ARMA was also concerned about the negative impact that publication of Dr. Prince's findings could have on its competitive position. ARMA's obligation was to convey safety-related information about its products to the Bureau promptly. It could have addressed risk, if any, of confusion by including in a timely report to the Bureau all the contradictory and inconsistent data it believed qualified Dr. Prince's findings. So that is the view of Mr. Justice Creever in his report. A criminal case was later brought in Canada against ARMA and against Dr. Riddell personally and against a number of other defendants as well. Part of the prosecution case was to allege that there had been a duty to disclose Dr. Prince's studies. The case was heard by Madam Justice Bonotto. She acquitted the defendants, and she came to a different conclusion to Mr. Justice Creever. In her view, ARMA and Dr. Riddell had been justified in their position, particularly in light of the other studies that the company had considered and commissioned. We could have MDUN 5020_250. This is paragraph 90 of Madam Justice Bonotto's judgment in the criminal trial in Canada. And I quote, "This was a time of great uncertainty. In the face of this, and in light of the clearly articulated studies of the CDC supported by the FDA, it would have been unreasonable, if not irresponsible, for ARMA to have thrust such confusing, incomplete, and inconclusive information into the community." Thank you. We can take that now. Madam Justice,
did she say anything about the central thesis in Ms. Justice Creever's comments, which was that there was a duty on ARMA to report the studies, whatever they were, to the Bureau?
Off the top of my head, I cannot remember if she specifically addresses that point. But what she does do is emphasise that she's coming to her conclusions not just on the criminal standard of proof, but she says that in her view, having listened to 1.5 years of evidence, the conduct of Dr. Riddell was reasonable, responsible, and professional. And Dr. Riddell himself was, and I quote, responsible and professional throughout, consistent with his well-deserved reputation for integrity.
I follow that, but if the suggestion is correct in what Ms. Justice Creever says, that the obligation on ARMA was to report to the Bureau the studies which they had available to them, then they should have sent all of them to the Bureau.
Yes, I think it's fair.
That would follow, I think, logically.
Yes, so Mr. Justice Creever's critique is about not providing it to the regulator.
Yes.
But Madam Justice Bonotto, when considering...
Is dealing with the conduct as opposed to the conduct apart from not sending it to the regulator, which they didn't do.
I think she is dealing generally with the allegation that was made by the prosecution that there was a duty to disclose this both to the regulator and to the...
What was the nature of the charge?
It was, I don't know I don't have the exact Canadian statutes to hand, but I can, if you give me a second, I will try to find the... So the charges were of criminal negligence and common nuisance, and they were brought against Armour, Dr. Riddell, the director of the Bureau of Biologics, the chief of the Blood Products Division and the national director of the Blood Products Service of the Canadian Red Cross. The corporation is also charged with, that's Armour, with failing to report a deficiency under the Food and Drug Act. Those accused were involved with manufacturing, licensing and distribution of heat-treated factorate for product.
So it was acquitted of a failure to report?
Well, the acquittal is of a charge of criminal negligence and common nuisance. And the part... I think we may need to have a look at that in due course, but thank you for that. I think it's fair to say that Madam Justice Bonotto deliberately stepped outside the legal test that she was required to apply in the criminal court in order to give her view that Dr. Riddell had behaved reasonably and responsibly throughout, including in respect of the Prince allegations. The presentation doesn't attempt to scale the mountain of evidence and opinion that has been formed in the wake of this controversy. Several of the protagonists have given extensive testimony, and indeed we've referred to the fact that the criminal trial, which this was one part, lasted over a year and a half. Previous inquiries have considered and commented on this matter. There are references to some of the documents and some of the inquiries in the written presentation. The technical details involved are complex. They deal with the sensitivity of assays, alleged arithmetic errors, and the questionable assumptions that underpin some of the work. I'm not going to try to go into that level of detail now. This is merely a preliminary sketch, as it were, of this issue and this controversy, which has been well documented elsewhere. And certainly, counsel to the inquiry do not make any comment on either Mr. Justice Krieger or Madam Justice Bonotto's view of matters. That concludes the presentation today and includes this round of presentations on the pharmaceutical companies. As I said at the start of these presentations, they are the result of a great deal of work done by other members of your legal team, and Ms. Richards and I are very grateful for the assistance that we have received from them.
Well, thank you very much indeed. That leaves us then with tomorrow we hear from Mr. Bishop, do we?
We do.
So 10 o'clock tomorrow. 10 o'clock.